What about inhibition in the CNS? As mentioned earlier, activation of inhibitory synapses results in a decreased likelihood of firing and action potential in postsynaptic cells. In the central nervous system, the vast majority of inhibitory postsynaptic potentials, or IPSPs, are due to the action of ligand-gated ion channels, including the GABA and glycine receptors. GABA receptors, named after their activating ligand gamma-aminobutyric acid, or GABA, are the primary IPSP-generating receptors in the brain. Glycine receptors, which bind the naturally occurring amino acid glycine, are the primary IPSP-generating receptors in the spinal cord. Structurally, these two receptor types are very similar to the nicotinic acetylcholine receptor we discussed earlier. The most relevant functional difference is the fact that the M2 domain in each receptor subunit is lined by either neutral or positively charged amino acids, and not negative charges, as in the case of the acetylcholine receptor. This is crucial since both GABA and glycine receptors are anion-selective ion channels, causing an increase in chloride conductance. The influx of chloride forces the postsynaptic neuron to stay close to the equilibrium potential of chloride, which is negative 65 millivolts, which is very close to the neuronal resting potential of negative 70 millivolts. So why go through all this trouble if nothing seemingly changes? While not as exciting, no pun intended, as IPSPs, inhibition is particularly important when a neuron is trying to avoid a go signal, yet is being attacked repeatedly by many weak IPSPs. As we'll see in an upcoming lesson, in a context when other excitatory neurotransmitters are trying to depolarize a single postsynaptic neuron simultaneously, inhibitory synapses can maintain a neuron at or close to resting potential. Such an inhibition mechanism is called shunting, and describes the fact that the negatively charged chloride influx can counterbalance the positively charged sodium influx of excitatory synapses, resulting in a stop signal. Pharmacologically, both blockers, or antagonists, and activators, or agonists, have been identified and developed for both glycine and GABA receptors. For example, strychnine, a common component of rat poison, is a very powerful antagonist of the glycine receptor. This blockade of the receptor causes muscular convulsions due to the loss of inhibitory action, and eventually it can lead to death through asphyxiation. Similarly, picotoxin, also known as coculin, is a poisonous plant toxin that can act as a channel blocker for the GABA receptor in the brain. As GABA itself is an inhibitory neurotransmitter, picotoxin has stimulant and convulsant effects, including muscle rigidity and seizures. Interestingly, picotoxin has effectively been used to counter barbiturate poisoning, which can occur during general anesthesia inside a hospital, or due to drug overdoses in recreational users. Barbiturates are drugs that act as central nervous system depressants by acting as activators of GABA receptors in the brain. They're believed to work through a mechanism called positive allosteric modulation. This means that the drug binds outside of the normal ligand binding site of the receptor, yet it has a positive modulatory effect on the receptor function when the normal ligand is present. Barbiturates in this way act by increasing the inhibitory effects of GABA on select pathways and circuits in the central nervous system. In today's medical practice, barbiturates have been largely replaced by more powerful drugs called benzodiazepines, sometimes colloquially called benzos. You might have heard of some of these drugs before, which include, for example, Valium and Xanax. Significantly less dangerous to overdose, benzodiazepines are, for example, used for the treatments of anxiety and insomnia. They also act by enhancing the effect of GABA at the central GABA receptor, resulting in a sedative, sleep-inducing, anti-anxiety, anti-convulsant, and muscle relaxant properties. Finally, alcohol is also known to modulate ionotropic GABA receptors to induce a calming and relaxing effect, within limits, but the mechanism of action is still poorly understood.